live right now. We will go live. Yeah, seems like everything is working well. Okay, so welcome to this second OoLEN public seminar. So we are live on both Zoom and on YouTube. And by the way, if you are an early career student or researcher working on the origin of life and are interested in joining OoLEN, you can go to oolen.org. There's a link where you can apply. It's great. It's a great community. We have a forum. We have discussions on many topics. And yeah, it's a great community. So today we have two presentations of about 25 minutes plus a 5 minutes Q&A. And for those attending on YouTube or even on Zoom, you can ask your questions verbally to the speakers. Or if you are on YouTube, you can ask your questions in the chat, and we are gonna relay these questions to the speakers after their presentation. So today we have two presentations, one from Gregoire d'Angers, and I hope the pronunciation is right, and from Peng. Okay. So first of all, we'll go with Gregoire, who is a professor of chemistry at the Aix-Marseille University. Gregoire d'Angers is also the leader of the Astro team at the Aix-Marseille University, and he's working on analytical chemistry, chemical reactivity, astrochemistry, astrobiology, and prebiotic chemistry. And today his talk will be titled From Astrochemistry to Prebiotic Chemistry, the Evolution of Organic Matter. So Gregoire, the floor is yours.
Okay, thank you very much for the presentation. Okay, so first, it's okay, you have? Yeah, I think it's okay.
Yeah, it's working.
So thank you very much for your invitation. So yeah, so today during this presentation, we'll try to see how astrochemistry can be interesting to understand the different steps that could have led to the formation of the first chemical systems and can then evolve to the emergence of life on the primitive Earth. So the main questions in which we are interested is that to understand what was the condition of the environment on the primitive Earth that allows the transition from the non-living to the living system. So as a chemist, we try to follow a very maybe simple scheme here is that we start from the identification of small molecules to try to understand in which environment these small molecules can lead to the formation of building blocks. So building blocks, if you look to the current living system, this is amino acid, nucleobases, sugars, and so on. And then we try to understand how these building blocks can associate, leading to the formation of macromolecules, and how these macromolecules can interact, leading to the first primitive cells, and so on, how this chemical system can evolve up to the emergence of the current living cells. So of course, the first step is to define where we can find these building blocks. So you have two different environments. The first environment corresponds to the environment of the primitive Earth itself. So you have the atmosphere, the volcanoes, hydrothermal vents, and the other source of organic matter, as we will see during these presentations, is the extraterrestrial bodies that are asteroids and comets. So which is interesting with this exogenous reservoir of organic matter is that if we compare to the endogenous one, is that we have a direct observable of the type of organics that were delivered at the time of the primitive Earth 4.3 billion years ago. Because we have meteorites on Earth, and meteorites are as parent bodies, asteroids and comets. So if you look to the organic content of specific meteorites that are carbonous meteorites, you can have a direct information of the type of molecules that were present of the primitive Earth 4.3 billion years ago. So if we look to these meteorites, you have about, for maybe up to 5% of their mass that were form of organics that are separate in 25% in soluble organic matter, and the rest is about 75%, which is named as insoluble organic matter. So, of course, because we are on Earth, we can use as analytical tools that present the best performance. So we can have a look on the molecular diversity of the soluble organic matter. So, for example, here I give you the results obtained by Philip-Schmidt-Coplins by analyzing the soluble organic matter with high-resolution mass spectrometry. So here you can see that you have the full mass spectrums, and you can zoom and zoom, and you go to the nominal mass, and you can see that you have numerous different ions that were detected. Which is important here is that for one ion that you have here in the Murchison meteorite, you can have up to 100 different isomers. So each peaks here have to multiply by the number of isomers. So that means that you have thousands of different molecules that form this soluble organic matter that you can find in meteorites. And because meteorites have parent bodies, asteroids and comets, you can say that you have the same sort of molecular diversity in these interplanetary bodies. So you have a huge molecular diversity. So of course, this is only a global view of the composition of this organic compound. So we can also target on specific molecules and molecules that can be interesting for primitive chemistry. So these are our building blocks. So you can see that in meteorites, you can observe sugars, nucleobases, as well as amino acids. So when you have meteorites that arrive on Earth, we know that we have the potentials of releasing some organics that can be really interesting for starting a primitive chemistry on the primitive Earth. So next to this soluble organic matter, you have an insoluble organic fraction, so which is more like a connection of aromatic structures with aliphatic bridge, and you have some heteroatom on this bridge. So this insoluble organic matter maybe is less interesting for primitive chemistry, even if some experiments have shown that if you put this insoluble organic matter in hydrothermal environments, you can release some specific molecules that can be interesting, like fatty acids, something like that. Okay, so maybe, yeah, it's not maybe directly interesting, but in some environment, maybe we can obtain some interesting molecule from the alteration of this system. Okay, so looking from this in this natural objects clearly show you that we have a very interesting molecular diversity that can be delivered at the surface of rocky planets. So now the question is that where come from this organic matter, this huge molecular diversity? So you have different hypotheses, but the main hypothesis is that all this organic matter will start by forming during the collapse of the dense molecular cloud. So you will have a first evolution that will lead to a pre-accretional evolution that will lead to the formation of organics that will be present of the different grains that will form the protoplanetary disk. And then you have a second step that leads to the formation of the different bodies of the planetary system, and of course of the solar system. You have the formation of planets that you erase all the organic contents, but you have the leftover of these accretions that are asteroids and comets. And as we've seen in these bodies, you have the trace of the primordial organic matter. But if you look on some of these bodies, we know that from the analysis of the mineral phase of some asteroids, we know that you have a secondary alteration that can occur in that body. And then, as we discuss, these bodies can go to the surface of planets, and then you can have a release of the organics, and you have a new evolution of these extraterrestrial organics, depending on the environment of the planet where it's released. So now we will try to see what sort of organisms can be formed in the first evolution, second evolution, and which would be the condition to obtain the emergence of the first chemical systems that will present the property of living system as we know them today on Earth. So the first step is to observe what are present in these dense molecular clouds and how it will evolve during this evolution from the solar nebula to the protoplanetary disk. So if you look in dense molecular clouds, so we have information of the composition of these clouds, and for 1% of their mass, they are formed of icy grains. So here you have the representation of icy grains. This is mainly a silicate core of some micrometers, and that are surrounded by an ice that is composed mainly of water, carbon dioxide, monoxides, methanol, ammonia, and some other compounds. So we can have a direct information of the composition of this ice by infrared spectrometry. Like a GWST telescope, for instance. And so when the dense molecular clouds collapse, you have these icy grains will evolve under high energy processings, and depending on their position to the burning star, they can be warmed, and so you will have an evolution of the first molecules that will lead to new molecules. Some of these molecules will be released in the gas phase, and some other, after the desorption of the main component of this ice, which is water, can be released on the grain and stack at the surface of the grains. So to follow this evolution, try to understand the reactivities that can occur in this evolution, we develop different experimental setups. So here you have one that we developed in Marseille. So here, the center of the system, this is vacuum chamber at 10 at -9 millibars. You have a cryogenic aid that allow to cool down the sample loader up to 20 down to 20 kelvins. You have different analytical tools that allow to follow the evolution of the ice during the processing and to analyze the gas phase and solid phase that can be generated. So now I will show you some very simple experiments that we perform to first identify what sort of molecular diversity can be really in the gas phase when you process an ice. So here we'll start from pure ice of methanol. So the experiment consists of forming this ice of methanol at 20 Kelvin at 10⁻⁹ millibars, and then we will irradiate it with UV at minimal alpha during 24 hours, and we will then warm the system that was processed, and we will recover the organic compounds that are maybe released in the gas phase during this experiment. So here, this is another view of the system. So you have the sample holder. We will condense at 20 Kelvin methanol that will form the ice. We irradiate with UV. We can have a direct information of the evolution of the ice during the irradiation with infrared spectrometry. And then after the UV processing, we warm the system and we will recover all the organic volatiles that will be released in the gas phase to be injected in the GCMS, gas chromatography, coupled to mass spectrometer to obtain their identification. So here you have what we observe with infrared spectrometry. So in red, you have the spectrum of pure methanols. In blue, after irradiation. So you can see that after irradiation, we can observe the formation of common species that are formed after the irradiation of methanol. So you have carbon dioxide, monoxide, formaldehyde, methane. And so as I told you, we then warm the system. So you can see here just that you have some small bands that appear in infrared spectroscopy, but with only infrared spectroscopy, it's really difficult to obtain information of this identification of these new molecules. So we warm the system and we will recover all the molecules that will be released in the gas phase. And as you can see, after the warming at the limit of detection of infrared spectroscopy, you do not see any organic molecules that are still present on the surface of the sample holder. So now what sort of information we obtained with GCMS analysis? So which is really interesting is that in fact, with this technique, we were able to identify lot of different molecules, up to 35 different molecules. And just remember that we only start from methanol. We irradiate it at very low temperature, low pressure, and you can see that you can generate a huge molecular diversity. So up to molecules with 6 carbon, you have, I think, all the different chemical functions that you can form from carbon, hydrogen, and oxygen. And all that you have in darks were the different molecules that we were able to identify with the GCMS, okay? And in blue were and pick the first molecules that were observed with other experiments. So this experiment, very simple experiment, show you that in that sort of environment, when you start to simulate the evolution of icy grains that are observed in this molecular cloud during the formation of a star, of a planetary system, you have a huge reactivity that occur and lead to a very important molecular diversity that can be observed from the gas phase. And why we obtain this important molecular diversity? This is because when you look to the ice, you have a combination of radical reactivities and thermal reactivity that lead to the formation of small organic molecules. And this reactivity increases when you add water. So water have a double roles. The first role is that it will desorb at 185 Kelvin in the labs, and that will drive a large part of the molecules that were formed initially in the ice in the gas phase. So this is what we observe here with the analysis of organic compounds with GCMS. But which is really interesting is that when you have the water can keep molecules at higher temperatures than their own temperature of desorption. That allows to the formation of more complex molecules. And when you have the desorption of water, you will concentrate the system, and that will lead to a higher reactivity that will lead to the formation of macromolecules. And that leads to what we call refractory organic residue. So now we've seen what sort of molecules were in the gas phase. Now we will try to see what sort of molecules remains on the sample holder after the desorption of water. So here you have a picture of what we obtain experimentally after the processing of an ice form of water, methanol, ammonia. Here again at low temperature, between 20 Kelvin or 77 Kelvin, depending on the experiment. Low pressure again. And you see, so you have here a microscope picture of the residue. So if it's clearly not a powder, but it's like a liquid and a liquid of organics here. And you can have spectroscopy information about molecular signature, chemical function. So here, this is a residue. We can obtain information on specific molecules that can be present in the system. So here you have hexamethylene tetramine. You have also polyoxymethylene that were observed by their specific bonds. But with X-ray spectrometry only, you can have information of the chemical function diversity that were present, but not really on the molecular diversity that are present. So that's why we develop a new approach that consists in recovering this residue formed from that sort of ice mixture in pure methanols, and we infuse this solution in high-resolution mass spectrometer. So in fact, this is the same approach that developed Fish-Schmidt-Coplin for the analysis of the soluble organic matter of meteorites. So we performed this experiment with Roland Thyssen at IPAG in France with an Orbitrap. And the first observation was that when you analyze this system, you identify thousands of different ions. Okay, so we verify that these ions come from the residue itself and not from contamination. So we fix that. And what we observe, in fact, is that here again, when you start only from water or methanol, ammonia at 20 kelvins, low pressure, you put energy to the system. UV radiation in this case, you warm the system. So you have the desorption of the volatiles and you have this residue that remains on the sample holder. You have thousands of different molecules that are formed with masses up to 4,000 atoms. And remember that you only start from 3 small molecules with only one source of carbon, which is the methanols. So that shows you that these ices are very interesting chemical reactors that lead to this high molecular diversity. So we have a really complex and rich chemistry that lead to a very interesting molecular diversity. So as for meteorites, and we can also target some specific chemical families, and particularly we try to see if we have our famous building blocks in the residue. So we search for nucleobases, and we check that we have nucleobases. You have also sugars that were observed. So this was performed by Cornelia Meinert in Nice in France. You have also Munoz Caro that observed the presence of amino acids, and some experiments from Kaiser also show that maybe some small peptides were present in this residue. So, okay, so now if we compare to what we observe in the soluble organic matter of meteorites, clearly we are able from IC in the lab to form something that presents similarities with what is observed in meteorites in terms of molecular diversity and specific chemical families. So of course, I do not say that we form what we observe in soluble in the meteorites, but we have something that starting from dense molecular ices lead to something that presents this similarity and very interesting similarity. But all the samples that I show you here are entirely soluble in polar solvents. So the question is that, is it possible from ices also to form this insoluble organic matter fraction? So we perform very simple experiments. So we took our residue. So here you have the picture of the surface of the residue. So here, this is a new representation of the molecular composition of our sample that is obtained with high-resolution mass spectrometer analysis. So with high-resolution mass spectrometry, you can obtain the stoichiometric formula attribution for each ions, and after you can obtain the elementary ratio HC versus OC, and we will use this mapping to compare the evolution of the samples. Okay, so here you have the samples before the over-irradiation, and to see if we can transform these samples to insoluble organic fraction, we only irradiate this sample after its formation at 300 Kelvin at 10⁻8 millibar with UV irradiation. Oh, sorry, it's not. And what you can see here clearly is that the surface of the samples, the morphology of the samples is totally changed. So here this was masked from the irradiation, and here it was irradiated, of course. And clearly we have a modification. And when you look now to the signature, the organic signature of these irradiated samples, clearly you can see that we have a totally transformation of the molecular content. We decrease in HC ratio, that means that we go to aromatization and we lose a lot of the oxygen content. And which is interesting is that you have formed this insoluble organic fraction. So that means that from these ICs, you can form soluble fraction and insoluble organic matter too. So now the question, okay, we form these samples in the lab. So as I told you, we have some similarities if we compare to the natural objects that are chondritic meteorites. So now the question is that if we directly compare these organic contents using exactly the same analytical techniques, do we have really similarity? So if you looked to what we obtained from the Paris meteorite, so we analyze the insoluble organic matter. If you compare, you can say, okay, we have quite the same repartition in the space regarding the molecular contents. But if you look to the soluble organic matter of the Paris meteorite, clearly this is totally different. Okay, so what we form as a soluble organic matter in the lab from ices is not what we observe in natural objects. So why? This is maybe only because here, what I show you, the results that we obtain in the labs are only simulation, experimental simulation regarding the pre-accretional evolution, the first stage. And what we know from the analysis of meteorites is that if you look to their barren bodies and mainly on asteroids, some of them have endurance secondary alteration, and for some of them, aqueous alteration. So what we perform now is that we took our residue from the ices, we put it in conditions, simulated conditions that could have occurred in barren bodies of meteorites. So we put in water at 150 degrees Celsius. And we look before and after this alteration. So here you have at the left the initial residue just formed from the ice processing, and after the alteration. And what you can see is that here again you have a total transformation of the organic content. Okay, we lose a lot of the oxygen content, and we start to maybe decrease the Hc ratio. So, okay, we have this transformation. So now we can try to compare again to the natural object. So here I superimpose the distribution of the Madison meteorites. So you can see that here again, clearly, this is totally different, okay, from the direct residue from the ice processing compared to the soluble organic matter content of meteorites. But if you compare to what we obtain after aqueous alteration, you can see that the residue tend to go to present much more similarities with what is observed in the Murchison meteorite about the soluble organic matter. So here again, I do not say that we form what we observe in the soluble in meteorites, but we have a scenario that start from ices of dense molecular clouds. We simulate the first evolution. We can so form analogs of the organic matter that maybe can be formed the surface of grains of this dense of this protoplanetary disk. So you can form soluble organic matter as well as insoluble one, and we can have a new evolution after the formation of the parent bodies of meteorites that tends to lead to the formation, to the evolution of organic matter that tends to present similarities to what we observe in meteorites. So this is really interesting. So now the question is that once this organic matter is delivered at the surface of planets and more specifically in the environment of the primitive Earth, is it possible that this organism, this molecular diversity can play a role in the emergence of life on Earth? Oh, so this is a new project that we start this year in Marseille. So we start from the observation of this exogenous organic matter. So you have a huge molecular diversity, which is purely from abiotic chemistry. Okay, so you have this molecular diversity that can be present on the primitive Earth. And now the question is that in which environmental condition we can use this molecular diversity for the development of a specific chemistry and the development of chemical systems that are capable of reproducing and evolving. And which is interesting, very important here to note is that to obtain this transition, this phase transition, we need to have an environment that will link this molecular diversity with a sufficient source of energy that will allow to the emergence of chemical systems that will present this specific property. And if you look to these chemical systems that we will call primitive chemistry, so the primitive chemistry for us is chemistry of chemical systems that are able to reproduce and to evolve. And if you have these two properties that emerge, then you have a continued evolution between this primitive chemistry and this biochemical emergence. And you do not have any frontier between both, because the Darwinian evolution will already start to apply to this evolution of this primitive chemical system. And you will have a continuous evolution up to and selection up to the emergence of the current life. Okay, so which is really important is that you can have organic matter. And now we know that you can form organic matter in a lot of different environments, in a lot of different astrophysical environments. And when you question the transition to the emergence of life, which is interesting, is not anymore maybe the organic content, but this is the environment in which this organic content will be present, because you need to have this sufficient amount of energy to obtain this phase transition. Okay. So now I finished. Okay, so I just to give you the conclusions that which is very important. This is now to try to understand what are the minimal parameters of the environment to obtain the emergence of this primitive chemistry, and to question in which extent this extraterrestrial organic matter can have served as a source of organic matter for the emergence of this system. Thank you very much.
Thank you very much, Gregoire. Wonderful talk. That was very interesting. Okay, do we have any question either in the Zoom meeting or on YouTube?
Yes, Zhen. Okay.
Go ahead.
Then go ahead.
I thought you had
if
a
you want to
question,
ask.
Nathan.
Yeah. I will have, but maybe Zhen has one first.
So any questions from YouTube, or I should start?
You can start. Yeah.
Let's see. Gregoire, could you kind of stop screen sharing?
Okay. Sorry.
Okay, sorry.
I have a question. I thought you had a question because you raised up your hand.
No,
so
it
Gregoire,
was applauding. So.
did you look at the irradiation dose influence on the complexity of the organic matter? And is there a proportional factor between the irradiation dose and the complexity of the organic matter?
Yeah, totally. In fact, so you have different ways of performing these experiments. You can make the co-deposition of your gas mixture with irradiation, okay, or you can make layer by layer irradiation. So we perform this experiment. So we make a layer of methanol and we make different time irradiation. So you increase the dose between 5 minutes to 24 hours, and we recover the volatiles. And clearly, what we see is that when you increase the duration of the irradiation, you increase the molecular complexity. But which is very interesting is that it depends on the molecular families. So what we observe, in fact, is that you form maybe after 5 minutes already up to 5 carbons molecules with esters or ketones, while for aldehyde or alcohol, you need to wait 15 to 30 minutes. So that will really depend on the chemical families because the reactivity, the chemical pathway are different. Okay, so yeah, so clearly this impacts a lot the molecular diversity that you can form.
Oh, great, thanks. Oskari has a question for you. What was the rate of falling meteorites with organic matter on early Earth? And there is a second question after.
Okay, so for that, I don't have the number in mind, but you have different publications that read about that, and I don't remember the name too, but I can send you if you want the reference about that. But as I remember, yeah, I don't remember. I will not do that. But you have different periods, which is interesting. Is the late heavy bombardment that occur between 4 billion years to 3.8 billion years, something like that. And here, because you have perturbation of the asteroidal center, that leads to a lot of asteroids that go in the internal parts of the solar system. And so you have a lot of asteroids that cross the Earth's environment and arrive on the Earth's surface. But I don't remember exactly the rate of deposition.
And so the second question into that is, would there have been enough to be relevant for the starting of life in terms of organic matter?
Yes, normally, yes. If you look to the simulation, the amount of organics that were delivered is quite important, okay? So, of course, it's always a debate between endogenous and exogenous, okay? But which is really interesting is that we have much more information of the organic content of this object that's on the real environment of the primitive Earth and the yields of organics that could have been generated in this endogenous environment. Okay, so that's why this is our starting point, because this is one observables, direct observables of the type of molecules that were present at the time of the primitive Earth. Okay, but after we can have a debate. If you have more reduced or more oxidized or more neutrals atmospheres of the primitive Earth, yeah, so you can have different yields. But this is you use this discussion, as you know, in the community.
And Anastasia has one question as well. Did you study how impact delivery affects the complexity of molecules and how if it changes the chemical structure?
Yeah, so you have experiments about that when they, so as I remember, it was published in Nature Zero Science. So yeah, they use a gun and they put ices or mixture of organics in the guns and they simulate the energies that could be input to the organic content during the impact or during the atmosphere entry. And what they observed, you can have some modification, okay? You can have some modification and some formation of molecules. Other experiments also were performed is that they performed space mission, okay? So they put organics in, I don't remember exactly, satellites that cross the atmosphere and they look to the evolution of the organic contents and the mineralogy, and they do not really found modification. But the main result, in fact, to answer to this question is that if you look to Hayabusa samples and that you compare to meteorites, you have quite the same organic content. The main difference is contamination because meteorites are contaminated with organics from biological system, which is not the case for asteroids. So what we know now is that when you look to meteorites, you have a very good insight on the organic content that you have on their parent bodies, so asteroids.
There are two more questions, but maybe then we present first and we will come back to questions at the end. What do you think, Alexandre?
Yeah, we could go on with Zhen's presentation and then go back.
I don't know. Yeah.
For Gregoire. I also had a few questions, but maybe we can ask them afterwards. All right, so we can proceed with Zhen Peng's presentation. So Zhen has been a member of OoLEN for more than 3 years. He's also a member of the executive board and the virtual meetings work group. He has a degree in biology from Fudan University in Shanghai, and then he also has a PhD from Washington University and has done a postdoc at Wisconsin-Madison, again in the US, and he's working on the origin of life, autocatalysis, complexity, and evolutionary biology. So his talk today will be titled Assessment of Stoichiometric Autocatalysis Across Element Groups. So Zhen, the floor is yours.
Okay. Thank you very much, Alex. So let's see. Today I'm going to talk about my recent study on the stoichiometric autocatalysis. I will explain what it is later across the periodic table of chemical elements. So let's first start with what is autocatalysis and why is autocatalysis important for the origin of life research. So let's start from two possible reactions. The first reaction is there are two molecules, F1 and F2, react slowly to generate products K and W. And in some other cases, maybe F1 and F2, if we put them together, they just do not directly react. But if a product of a reaction, let's say K, can catalyze the reaction, then we have a case where the product of a reaction catalyzes the reaction, producing the effect that this product consumes the reactants as food and propagate more of itself. So this is generally called autocatalysis. And we can view this process in another form, that is, we put a K into a reaction, react with F1 and F2, which are foods, generate more Ks and some waste, which is W. And why is autocatalysis important for origin of life? Well, you might already realize that the process mimics reproduction or propagation. So there were two conditions or two features of autocatalysis. The first one is stoichiometric condition, which exactly describes this feature. You have some autocatalysis, let's say AK, react with some food, BF, generate more of K, and maybe there was some W. And to make this process more look like reproduction, we also need to make sure that the direct reaction among food is very slow or never happens. Otherwise, this spontaneous reaction between foods will obscure or mask the stoichiometric autocatalytic process. And then we already know that within biotic or prebiotic chemistry, there are many cases of autocatalysis. The most famous one maybe is the oxidation of oxalic acid by permanganate, which you might have learned at high school or even university. This reaction generates a product that is manganese ions, and manganese ions can catalyze the process. So this is autocatalytic. And we also have a really famous example. It's a foremost reaction where the formaldehyde can react to generate some glycoaldehyde and other monosaccharides. And the core process of this foremost reaction can be written as that the glycoaldehyde catalyzes the formation of itself from two molecules of formaldehyde. On the other hand, we also have very, very many cases of autocatalysis in life. And in fact, every propagation process in life is a special form of autocatalysis. So cell catalyzes the conversion of from nutrients to more cells and some waste. And virus consumes living cells as food to generate more viruses and dead cells. So there's clearly some parallel between abiotic autocatalysis and biotic autocatalysis, but can they be even more similar? Well, let me give you some examples. We know that one autocatalytic cycle mimics the propagation of one species or one population. Then if we put two autocatalytic cycles together, maybe they can mimic ecological interactions among species or populations. So this is exactly the case. The left panel shows an example where the blue autocatalytic cycle consumes the orange autocatalytic cycle as food to propagate itself. And if we simulate its dynamics with some specific parameter settings of the reaction rate constants and food concentrations provided in the environment, we will see the oscillation, which is very famous for the predator-prey interactions and was first modeled by the Lotka-Volterra model. And on the other side, this is also a famous example in ecology, where the lynx eats hare in the forest. And then we also observe the oscillation of the prey and predators over a long period of time. So this is predator-prey interactions, abiotic and biotic. And we can also find some interesting examples in abiotic autocatalytic systems of mutualism. So here the two autocatalytic cycles, the blue ones generate a waste called Y, and this Y is actually a food of the orange autocatalytic cycle, and the orange autocatalytic cycle happens to generate an X, which is also a food of Y. So these two are mutually beneficial or mutualistic. On the other side, we know that there were some interactions between animals and the gut microbiota in its digestive system. So animals produce some waste which can be consumed and utilized by the gut microbiota to make some vitamins which are actually waste for the microbiota, but very beneficial for animals. So this is also mutualism. And with all of these, it will be interesting if we can assess how many autocatalytic cycles are there in an abiotic world, because it is generally considered that autocatalysis is a very special phenomenon in the world, so that it might be really rare and the origin of life might be a really hard process. And the second question is, for now we know that the autocatalysis in the life as we know it are based on carbon, but are there enough number of autocatalytic cycles which are not carbon-based that might be utilized to generate other lifelike systems, which may be an alternative form of life on another planet or other worlds? Well, these two questions are the main motivations for me to do an assessment of autocatalytic cycles across element groups in a periodic table. Let's start from a very simple example, and actually one of the earliest examples of other classes we know in history. This is some quotes from William Oswald, who first proposed the concept of other classes. This case is putting a piece of copper into nitric acid, and according to his description, this process will be catalyzed by the nitrous acid, which is actually a product of the process of reduction of nitric acid by copper in an acidic environment. So the dynamics of this process is a sigmoidal process, meaning that at a first step, the reaction is very slow, but as there were more nitrous acid formed, the reaction goes faster and faster. And at last, since the food here, the copper and the nitric acid, and also the hydrogen ions are fewer and fewer, the reaction goes slower and slower, goes to a plateau or a platform. And so if we analyze this process, it is actually formed by two sub-processes. The first one is a reaction where the nitrous acid react with nitric acid to generate two copies of nitrogen dioxide and water. And the second process is nitrogen dioxide react with copper and the hydrogen ions, generate two copies of nitrous acid. And then you have two copies here, you have two reactions here. And as these two processes goes on and on, we have more and more nitrous acid and nitrogen dioxide. And also, of course, the water as waste. And the first reaction is actually called proportionation in general, where the nitrous acid has the nitrogen in nitrous acid has an oxidation number of 3, and the nitrogen in nitric acid has an oxidation number of 5. And then react to generate two copies of an intermediate oxidation state nitrogen dioxide with 2 copies. So proportionation can be generally understood as a reduced form plus an oxidized form generate more copies of intermediate form. And then this intermediate form gets reduced by copper and goes back to the reduced form of nitrogen, is nitrous acid. And let us consider another case. Say we still have this proportionation process, but this time we oxidize this intermediate state nitrogen by some oxidant. I don't know what it is, but let's just assume that there is one. Then it will goes back to the more oxidized form, that is nitric acid, which also form another catalytic cycle. So in fact, there were two forms of this type of proportionation-based autocatalytic cycles. One is oxidative, just as I mentioned. You use some oxidant to oxidize the intermediate state back to the more oxidized state and consumes the reduced form to generate more of the intermediate state. Or you couple the reduction process and proportionation process to reduce the intermediate state back to the reduced state and consumes the oxidized state as food. It goes on and on. So this just deal with the stoichiometric condition of autocatalysis. So that is why I say assessment of stoichiometric autocatalysis. The next question is how to find these kind of proportionation-based autocatalytic cycles. Well, the first step is, of course, literature. There are some really famous examples of autocatalytic cycles which were not explicitly mentioned as proportionation-based, but after I analyzed them, I found there are. This is an example of chloride iodide reaction, and I'll just focus on one example here. Let's take a look at this purple dots mention the reaction goes. Let us look at the reverse direction. It's HOI react with the iodide ions, goes to I2 is iodine. So there were two iodine atoms in the iodine molecules. And then this one can be, let's see, oxidized by the chlorine to 2 copies of HOI. And this HOI can go back to here, consumes the iodine ions, and this process can go on and on. This is autocatalysis. And we also have other similar examples. Here is if we let the ion 2 ions react with mercury. You can have autocatalytic process. And you will also see the example, just as I mentioned, the oxidation of oxalic acid by permanganate. This is an example of comp AC. And then another famous example is called the BZ reaction, Belozov-Zhabotinsky reaction. It actually consists of two comproportionation-based autocatalytic cycles. So there are many cases there. Then I'm thinking whether I can really compile as many of these examples as possible to see whether this phenomena is a general or a special case. And while I was searching for these examples, I found an interesting thing that the comproportionation, this term, was not only used in a form that I just mentioned. Sometimes it's used to describe a reaction where the change of the oxidation number does not follow the high plus low goes to intermediate. For example, this reaction, O2SCl2 plus O2SF2 goes to 2 copies of O2SFCl. This is also mentioned as comproportionation, but the oxidation numbers of the elements does not change before or after the reaction. So it is also used in this format. I call it broad sense. It's rarely used, but it's nonetheless there. And actually, there are some other examples of autocatalysis that matches this definition of broad sense comproportionation. This example is the oxidation of, oxidation of pyrite by oxygen catalyzed by ferric ion and ferrous ions. So this matches the broad sense comproportionation. It's a redox reaction, but the key process here is not high plus low goes to low. It's just low plus high goes to low, because the sulfur here provides the electrons. And the famous Formos reaction also matches the broad sense comp ACs. The oxidation number of the carbon in general does not change before and after the comproportionation, but the process is nonetheless autocatalysis. And I searched for literature to check how many comproportionation reactions I can find. And some of these literature are not written in English or Chinese, so I can use some machine learning based translator online and technical dictionaries to check whether they really mention that. And I strongly recommend this resource. It's very useful. And here is an example of the earliest literature I found. It's from 1832 by Heinrich Rose, written in German. I could not understand German, but after I type in this thing to the online translator, I can understand that it describes a comproportionation of the phosphorus element. And the next step is to find the auxiliary reactions, as I just mentioned before, the reduction reaction or oxidation reaction, according to the documented comproportionation reactions from literature. And the third step is to check whether the stoichiometrics of the reactions I just found can match the definition of comproportionation-based autocatalytic cycles. And I also add the original text to my database. And here is what the database looks like. You will find this as a supporting information in my publication on JAX. And so this panel shows which element it is, which element goes into the comproportionation process and which group it is in the periodic table. These are the reaction equations, the source of the literature, and some quotes from the literature to help me and also help readers to identify exactly where the reaction equation is mentioned, because sometimes the reaction equations requires special reaction conditions to occur. And if someone wants to experiment, they might want to locate exactly where the reaction is mentioned. And last, I find over 200 compases and over 44 broad sense compases. And these are much more there are likely much more cases, but I decided to stop there because these are already many and are enough to say that broad sense and also non-broad sense comp ACs are a general phenomenon. If we look through the periodic table, each of the element groups or the lanthanide series or actinoid series have at least 2 comp ACs as examples. And most of these comp ACs have no more than 3 reaction equations in it, meaning that it might not be that hard to make all the reactions to occur to form another catalytic cycle. So we can conclude that the other comproportionation-based autocatalytic cycle represent the general phenomenon across the periodic table. And as I mentioned, there could be ecological interactions between autocatalytic cycles. Of course, there can be larger interactions between comp ACs. And here are just some special cases. Not all the cases, but I think they're interesting. There can be competition between comp ACs. When both of them are oxidative comp ACs, they might compete for the shared oxidant. And they can be mutualistic. If one is oxidative comp AC, the other is reductive comp AC, so it can recycle a reductant oxidant pair. And the mutualism might be even more strong if the auxiliary process of an oxidative comp AC happens to be the auxiliary process of the reduction of a reductive comp AC. And they can, of course, have predator-prey interactions if an autocatalyst of one comp AC is the food of another comp AC, like this and this. And there can also be bistability if the intermediate state of autocatalyst of one comp AC happens to dimerize with the intermediate autocatalyst of another comp AC to form a dimer. So with all of these examples, these are good theoretical. Can we find examples in the real world? Well, yes. Here's an example, just I quoted from the previous example of comproportion of chloride iodide reaction. This is a comproportionation of iodine. This is a comproportionation of chlorine. And then this reaction happens to be the auxiliary reaction of both interactions. And we can also, as I mentioned, there are interactions in the Belousov-Jabotinsky reaction, where there were 2 comproportionation-based autocatalytic cycles, and the autocatalyst of one is the food of the other. And as I mentioned, we confirmed that the stoichiometric autocatalysis can be represented by comproportionation-based autocatalytic cycles. But we still left one thing behind, that is the kinetic condition of reaction equation. We want the reaction between the food for comp ACs, the oxidative food and reductive food, to be very slow or even never occur. And how to make this happen? Well, there were, but if the reaction is very fast, and then this direct reaction between food is fast, and the indirect reaction between food catalyzed by the autocatalyst is also fast, then the spontaneous direct reaction might mask or obscure the autocatalytic reaction. So even if there is one, we might not be able to observe it. And how to facilitate our observation of the autocatalytic dynamics? Well, we can kinetically, spatially, or temporally separate the food species to observe a typical autocatalytic dynamic. And by kinetic separation, I mean that the foods, they may freely contact, but whenever they contact, they just do not react fast enough to form the product. And this is actually covers the most examples we know about autocatalytic process, because people usually do experiments in a well-mixed reactor, even either closed or a flow reactor. And the famous examples, abiotic examples, as I mentioned, the BZ reaction, chloride iodide reaction, and permanganate oxalate reaction. And in the biotic world, we have the Kelvin cycle, which is utilized by the green plants to synthesize monosaccharides from reductive force, the NADPH, and carbon dioxide, and also water. And there is also an example called reverse triboxylic acid cycle. It can also synthesize small organics from inorganic molecules. And by spatial separation, I mean that in some cases, the food of the comproportionation process and the food of the auxiliary process are separated by some spatial barriers. Let's consider this autocatalytic cycle. The carbon dioxide react with carbon powders to generate 2 copies of carbon monoxide, and this is a gas diffused through the tunnel to another reactor where it reacts with iodine oxide, and this carbon monoxide is oxidized to carbon dioxide, which is also a gas, and diffused back to this right chamber, which is very hot, and react with the carbon again. So here, the carbon powder and the I2O5 powders are separated by the spatial barrier. And there are abiotic barriers that can be utilized to do this job. For example, the water bodies can separate the food in the atmosphere and the food in the bottom of the ocean or lake. And mountains can separate rocks in different geographical regions. And the space between rocks and ores, for example, maybe some airs, atmospheres, or even oils may separate food from different rocks. And then the biotic barriers can be utilized, include intracellular compartments, for example, the membrane-based organelles in cells, and also the migration process can also be understood as a form of biotic spatial separation, because fish and birds, they might need different foods, different temperature, and different environments in their different stages of a life cycle. And finally, the temporal separation, this is kind of like a mechanism I refer to it, because we just need to provide foods at different different time intervals for the either the comproportionation reaction or the auxiliary reaction. And then we can see the propagation of the other catalyst. For example, the core comproportionation process here is H2S react with sulfur dioxide SO2 to generate more copies of S. And then later, we add some oxygen back to the reactor, and it will oxidize the sulfur back to SO2. And this process goes on and on. We will see the propagation of SO2 and sulfur, elemental sulfur. And the mechanisms that can be utilized to do abiotic temporal separation, including tidal waves, diurnal cycles, geyser eruptions, and intermittent rainings or flushings of the water on the surface of rocks. And then the biotic mechanism for temporal separation, including vegetative and reproductive growth in plants. So for plants, sometimes they usually do vegetative growth in the early stage of life, and later they transfer the resources or utilize other resources to do the reproductive growth. So the food are provided and consumed at different stages. And also circadian rhythm are utilized by some species to consume different foods, because maybe in the morning you might have a lot of sunlight, and in the night you might be able to have a lower temperature, which help you to go against the vaporization of water. And this is actually used by many plants in the regions like desert. So with these 3 mechanisms, you can more easily observe autocatalytic cycles. So in conclusion, autocatalysis is very likely a very pervasive phenomenon across the periodic table, not just about carbon, but about many different elements, not a collection of special cases. And non-carbon-based complexes suggest that it is possible to have alternative forms of life or extraterrestrial life and lifelike systems. And the possible existence of the stoichiometric autocatalytic cycles does not mean that they can be easily observed. To observe the typical autocatalytic dynamics, kinetic, spatial, or temporal separations between foods will be necessary. And here are some future directions I'm going to do in the next several years. I want to add more complexes to the database add more parameters to the databases, for example, the rate constants and reaction conditions, to help select systems that I can do some experiment on, and find other general mechanisms of ACs except for comp ACs, and also rank the comp ACs according to the easiness of experimental testing. So I can create some lifelike systems in lab. And finally, I would like to thank my co-authors, Zach, Albert, and Batul. And I also got some very helpful comments from Dr. Baum and my colleagues Bruno, Evrim, and also a scholar from currently working in France, Wu Tao. And I would also thank funding from also the private section and also the government-supported section from both the US and Australia. And finally, thank you very much. And you can check more of the details in these publications. And any questions?
Thank you very much, Zhen. Very interesting. Yeah, we'll take any question now. So we have a question from Oskari Lehtinen in the chat. Okay, question on the autocatalytic networks. How could the network survive against parasitic reactions that would feed on the network substrates, but not contribute to the network?
Okay, so this actually relates to the kinetic aspect of the reaction. It all resides in the values of rate constants. And that is actually the kind of the rabbit hole there. If the parasitic reaction or parasitic process or parasitic autocatalytic cycles has very high rate constants, then it might drive the extinction of both the host one and the parasitic one, because whenever you generate some small amount of autocatalysts of the host autocatalytic cycle, it might be really fastly consumed by the parasitic one. And the next, but if the parasitic autocatalytic cycle does not have that fast reaction rate, the rate constants, then it might actually co-survive with the host cycle. And what's more interesting is that if the parasitic cycle actually consumes food from multiple autocatalytic cycles, it can actually increase the diversity of its hosts, or actually it can be viewed as prey. This is similar to what we observe in the ecological interactions, where some herbivores, they compete with each other for the plants, but and then in some cases, one herbivore consumes the plants much faster than the other. But if we put some carnivores one carnivore into a system, the carnivore could maintain a balance between the two herbivores. So actually, the two herbivores can coexist with the carnivore. And a similar process may also happen in the abiotic world, where when you have multiple autocatalytic cycles, they might be consumed by the shared parasitic or predator cycle, and that could increase the diversity. And in some cases, the predator cycle might actually give some benefits to its host. Let me give an example here. It's not mentioned in detail. Let's see. Here, chloride iodide reaction, one of my favorite systems. So you can see there are multiple curves here. It means that the interactions between all of these autocatalytic cycles are complicated. And then in some cases, I could not remember which cycle is there, but one cycle here consumes the autocatalyst of another cycle, but the waste generated by that this predator autocatalytic cycle happens to be an autocatalyst of the prey autocatalytic cycle. So the relationships are very complicated, and it's just for any specific cases, we need to analyze specifically, and that's it.
Another question from Marina. In your opinion, can we make important advances in the origin of life following Kaufman approach, or or we really need to take into account the chemical details of autocatalytic cycles?
Wait a second.
It's in the Zoom, in the YouTube live.
YouTube,
So
okay.
in contrast of Kaufman theory of autocatalytic
Okay,
sets
so
remains much
Kaufman,
more abstract.
that is something. The answer would say this is a very good question, but the answer could be very complicated. So Kaufman's original paper about autocatalytic sets was published in 1980s. I believe it's 1986 or 1987. He used the peptide network as an example, and that was in fact the autocatalytic sets proposed by Kaufman in 1980s is fully compatible with the broad sense comproportionation cycle, because it is conceptually just similar to the Helmholtz reaction if we view these formaldehyde as amino acid residues. But later, I think he published some paper on something called reflexively autocatalytic sets. It's called RAF sets, and some cases of the RAF sets are not compatible with the comp-ACs I propose here. And I'm still kind of investigating what causes the difference. But in general, both cases can give rise to complex dynamics among autocatalytic networks. And I can find some ecological interactions in their networks, although they did not explicitly mention that. So that is the answer I can currently give. And yeah, the definition of autocatalysis is definitely a rabbit hole. If you check my paper about, let's see which one. Yeah, if you check this one, actually write this with other OoLEN members, Klaus and Joanna, bioacids. We briefly reviewed the early history of the concept of autocatalysis and found that what we commonly understand as autocatalysis today is just a proper subset of what proposed by William Ostwald. So yeah, hope that will be a satisfactory answer.
I have one question for you. It will be dependent on the ecological and chemistry autocatalysis way, but for the chemistry part, when you say it has to be a through kinetics, how slow, is there a threshold or something that can be put on, or at least a range where we can say it's slow enough to be considered?
So it's about, yeah, let me think. It's a good question. I need a few seconds to think about how to organize my answer. And so about ecological interactions. Yeah, I think the short answer would be for specific cases, I need to use specific analysis. I try to find some general principles that can be used to infer what type of ecological interactions are there in a network, but the results are always complexity over complexity, and I cannot find a general phenomenon. And that bugs me really much. And
yeah, no, it was much more like when you say fast, like when can we consider that it's too fast?
Yeah, it's not just about rate constants, it's about reaction conditions. The high temperature and low temperatures are different. If you think about the autocatalytic process of something called tin pat, like the famous Napoleon's story where the buttons were made by tin, and when they're put in the cold weather, the autocatalytic process of converting the shining tin into the powder tin goes very fast. and then you have the buttons goes into powder, and they cannot protect them from the cold winter. And then, but if the temperature changes, this process is inhibited, and also the concentrations of the food chemicals will matter really much. Even for a autocatalytic cycle where the rate constants of the autocatalytic direction is not large enough, you can still have that cycle survive if you provide enough concentration of food chemicals. And then you can even increase the concentrations more by increasing the pressure of the system. So all of these factors are just interweaving with each other, and I'm still trying to find a way to disentangle them.
Thanks. Another question from Oscari. How do the reaction networks sustain themselves in the presence of all the waste products? And will not the reaction space get filled with waste at some point?
Well, the waste can be, this is a good question because it gets correct that if you have a lot of waste products and consider reversible reactions, then the forward reaction, the direction will be largely inhibited by the waste. But the waste can be removed in multiple ways. For example, removed as precipitant, removed as by ventilation, or removed by being consumed as food by some other autocatalytic cycles or other processes. So that is a way we can do that. And if you think about the history of the evolutionary history in the biotic world of biospheres, there are many cases where you have like corpse or waste that could not be consumed through a long period of time, but later in evolution gives you a new species that consumes it. The famous example might be the coal, right? The first time when you have kind of tree-like plants on the planet, the bacteria and fungi could not consume the wood, and that is why their corpse goes into the coal. But later, when the bacteria and fungi obtain the ability, then the era of forming the coal was stopped. And then these like termites and all of these kinds of small organisms start to consume the waste or the corpse of the previous autocatalytic cycles. And I think similar things also happen in the abiotic world. And I think, do we still have enough time to go back to the question for
maybe for Gregoire? Yes. Gregoire, do you have some time for 2 more questions? So the first question is, what do you think about the idea of aqueous alteration not being a secondary process acting on ice chemistry products, but aqueous chemistry being a primary synthesis pathway of organics in meteorite parent bodies?
Yeah. Okay.
Yes.
So you have different analysis of the content of meteorites that shows that organics and water ice were incorporated during the accretion. So after the question is that where come from, what sort of small molecules you will incorporate in your parent bodies to form organics chemistry? So that's why I don't really understand the questions. Because okay, you can make aqueous chemistry, but you have to start from you will have minerals, but you have also to have an organic content. So I don't know if you estimate that you can incorporate carbon dioxide, methane, or something like that, and then you can perform experiments inside the parent body. So maybe you can clarify this point. But yeah, for analysis of meteorites, clearly you have some trends that lead to things that you already incorporate organic matter inside the system. And if you look to isotope analysis, you can see that you have higher isotope are higher present higher abundance in meteorites. So that suggests that some of the organics that are present come from low temperature chemistry. And if you start from low temperature chemistry, you probably have ice chemistry also incorporated inside that. So, okay, I do not say that all organics come from before accretion, and you have a reprocessing. This is our points, is that probably you have a lot of organics that are formed during the solar nebula evolution. So we focus on the solid phase, but you have the gas phase that can form maybe more insoluble also organic matter that can then is incorporated in parent bodies, and then you have a reprocessing in the parent body of meteorites. Yeah, okay.
Yeah, I was wondering about, yeah, starting from simple volatiles,
Yeah,
carbon dioxide.
So maybe, yeah, sure. And maybe you can have a part of the organics that come from that, maybe with Fischer-Tropsch reaction, something like that, depending on the temperature and the minerals.
And the second question was, did you discover any symmetry breaking effects or any homochiral compounds?
Yeah, so you do not have homochiral compounds, but which is interesting is that if you look to amino acids that are non-biologic, okay, so because the main problem of meteorites that's you can have contamination of biological system. But if you look, for instance, on isovaline, that is normally not present in the main part of the biochemical system, you have an enantiomeric excess up to 20% in L form. And the main question is that where come from this enantiomeric excess? So you have one scenario, you have other scenario, but one which was tested in the lab is that you can use UV-CPL irradiation of the ice. And when you pair from that, you form enantiomeric excess up to 2% of L form or D form, depending on the orientation of the UV-CPL. And then you can maybe have an amplification of this enantiomeric excess inside parent bodies during aqueous alteration. So this is what we try to investigate now, is that during this alteration, we see that we have interaction between sugar, amino acid, and so on. And so we try to see if we can amplify this enantiomeric excess up to what is observed in meteorites. But yeah, so you have an enantiomeric excess, you do not have homochiral compounds, and we try to understand how this enantiomeric excess emerge. And the last point is that so you have scenarios that present this enantiomeric excess as a possible imbalance in the organics that was present on the primitive Earth, and that maybe drive a chemistry that amplifies this system and go to the homochilarity at the end, because homochilarity is quite simple. This is only because this is the most stable systems, and you have the higher catalysis effects that you obtain in enzyme. So yeah.
Thank you.
Do we have more questions on YouTube? No, I don't think so. Feel free to ask any other question, either to Gregoire or Zhen.
Maybe if I can, just one announcement, because if you are interested, we have position in our team, or PhD position in our team, already primitive chemistry lab development and astrochemistry also. So if you are interested, do not hesitate to contact us. Okay, we have 2 postdoc position and 1 PhD position that are available for 2024. Okay, so do not hesitate to contact us.
Interesting. Great. Thank you, Gregoire. Okay, so thank you to both of our speakers for today. Wonderful talks, very interesting. And thank you, David, for the moderation. And thanks again to everyone who attended today's meeting, both on Zoom and YouTube.
Thanks to you for the invitation.
You're welcome.
Thank you.
Okay.
Thanks.